Hello everyone, welcome to a new session on Introduction to Bioinformatics, Importance and Use of Bioinformatics. Importance of Bioinformatics. Bioinformatics requires people from different working areas as it is a multidisciplinary field. It is the combination of biology and computer science and is a new emerging field that helps in collecting, linking and manipulating different types of biological information to discover new biological insight. Before the appearance of bioinformatics, the scientists working in different biological fields such as human science, ecological science and many other fields were in a necessity of some tool that helps them to work together. All the researchers in biological field are interlinked and had important information to share each other, but the problem was how to integrate. In such circumstances, bioinformatics emerges to help these scientists or researchers in fast research and leads to quick inventions by providing readily available information with the help of computer technology. Scientists and researchers spend their whole life in inventing things for human benefits. After so many years of development, they have collected huge amount of valuable data from their experiments, and the collection of data is still continuing for the better understandings of human life. Problem arises when they need to repeat the research just because the old data is hard to obtain or they do not know whether it exists or not. This wastes their valuable time. Let us take an example of DNA identification. Every species or human beings have particular DNA strands that contain the genetic instructions used in the development and functioning of all known living organisms. By identifying DNA information, one can trace generations, links and can find the root of different disease. Earlier it was hard to manage this information. In order to collect and link DNA information from all over the world and to solve many medical complications, bioinformatics is a very helpful hand for them. In addition, scientists also need a tool that can interlink information from different areas like biology, statistics, genomics, etc. to make their research faster. For instance, they may need some data regarding effects of particular gene on human beings and its effect on animals or on other species, so that they can interlink and generate some beneficial results or antidote that helps in human development. Eventually, bioinformatics provides that help in interlinking information from different fields and leads to quick results. Finally, bioinformatics also helps in digitizing the information available on the paper or in the form of specimen, so that with the help of internet it could be easily available to everyone everywhere. These days, computer is an important part of every research, without which so fast development cannot be imagined. Moreover, these days it is important to keep everyone aware of current developments. This will enable everyone to enhance the academic and research skills in their fields at minimum expense of time, money and matters. In this scenario, bioinformatics makes information readily available by collecting, linking and manipulating. Briefly, bioinformatics is playing a vital role in development of society by providing quick information and making research faster. It is using today's computer technology and biological research together very efficiently. This field is going to generate more opportunities in future for all people working in different areas. As you can see, the present challenges facing bioinformaticians is to improve database design, develop better software for database access or manipulation, and devise data entry procedures to compensate for the varied computer procedures and systems used in different laboratories. Today's role for most bioinformatics companies is to try to pull these labs away from their paper-based reporting systems and move them electronic, web-based systems. Adding, storing, retrieving and sharing information becomes easy once the data is made electronic. Use of bioinformatics. As being an interface between modern biology and informatics, bioinformatics involve discovery, development and implementation of computational algorithms and software tools that help us to get a clear picture of biological processes. Bioinformatics have its own importance in agriculture also. In developing country like India, it plays a big role in agriculture where it can be used to increase the nutritional content, to increase the production, to get a resistance against diseases, etc. In pharmaceutical area, bioinformatics helps to reduce the time and cost involved for different analysis, drug discovery, etc. The use of bioinformatics is threefold. First is to collect and organize the data in a way that allows researchers to access the information and to enter new results as it is produced. Just depositing these data into a database is useless unless it gets analyzed. So, the second aim is to develop tools to analyze this data in required way. For example, it is of interest to compare a sequenced protein with the already existing sequence may need a tool or program for the purpose of comparison. Developing this kind of resources can only be done with a deep understanding of computational theory and biology. The third aim is to analyze these data using these tools and interpret the results in a biologically meaningful manner. In the traditional biology, it is impossible to examine the result of an experiment in an extendable way. But using bioinformatics, we can conduct a global analysis of all the available data. The experiment results from biological laboratory will be stored into the bioinformatics databases, and then the analysis of these data can be done by using different computational techniques and tools. The data will be analyzed in terms of sequence, structure, function, evolution, pathways, etc. The result of analysis will be then compared with the biological data and the experiment result. Let us see these aims in detail. The first and core aim is the collection of information or data. So, what does this information stand for? Data can be raw DNA sequences, protein sequences, macromolecular structures, genome sequences, etc. DNA sequences are strings with 4 base letters A, T, G and C, which comprises genes. Next one is protein sequences. These are nothing but a collection of 20 amino acid letters. The most complex form of information is the macromolecular structural data. More than half percentage of this data is the structure of proteins. Genome sequence information is the collection of raw DNA sequences ranging from 1.6 million bases to 3 billion bases. Now, the issue is with the organization of this large amount of data. First of all, most of this data can be grouped together based on the biological similarities. For example, genes can be grouped together according to their function, pathway, etc. So, the major aspects of managing this data is developing methods to assess the similarities between these biomolecules and identifying those that are related. There are different databases available to deposit this data according to the information it carries. Each database contains different types of data. Some examples for the database are protein sequence database, nucleotide sequence database, structural database, etc. So, according to the types of data, these can be stored in different databases. Now, the question is that how can we use or analyze this data in practical life? The information stored in databases is meaningless unless it gets analyzed. There is different type of analysis can be done using these data sets. The raw DNA sequence data can be analyzed to find the coding and non-coding regions of the DNA sequence. Protein sequences can be compared using bioinformatics algorithms. This method is using for multiple sequence alignment. Annotation and encoding of sequence is the way to explain a whole genome. As we mentioned before, our body contains lots of proteins, genes, etc. The relationship between these proteins, biological pathways of different genes, details about the gene products are the core research areas in bioinformatics. One of the major areas of research is at the gene expression level. The gene expression level research is a way to understand the protein and mRNA that are produced by the cell. The main application of this research is in the area of cancer. As we all know that the reason and cure of cancer is still unknown, bioinformatics algorithms can find the shared sequences between different genes or proteins. The research on gene expression is trying to solve these questions. The use of bioinformatics depends on the area or field we use it. Moreover, bioinformatics has its own importance and use in every field of study. Some uses of bioinformatics is explained here. Depends on the branch of study. Structural bioinformatics. The branch of bioinformatics that deal with the analysis and prediction of the three-dimensional structure of biological macromolecules such as proteins, RNA and DNA is known as structural bioinformatics. It deals with generalizations about macromolecular 3D structure such as comparisons of overall folds and local motifs, principles of molecular folding, evolution and binding interactions, and structure of function relationships, working both from experimentally solved structures and from computational models. The structural bioinformatics can be seen as a part of computational structural biology. Structural bioinformatics has existed in some form or the other ever since the determination of the first myoglobin structure. Structural bioinformatics has a significant role in the field of molecular biology. The main challenge for structural bioinformatics is the integration of structural information with other biological information to have a deep understanding of biological function. The success of genome sequencing projects has created information about all the structures that are present in individual organisms as well as both the shared and unique features of these organisms. Homology models for the structures identified through genome sequencing projects can be created with the help of structural bioinformatics. The resulting structures will be studied with respect to how they interact and perform their functions. Similarly, the emergence of microarray expression measurements provides an ability to consider how the expression of macromolecular structures is regulated at a structural level, including the reactions associated with transcription, translation and degradation. The alteration in functional characteristics such as structure variation, genetic mutation and post-translational modifications can be studied through structure modeling. Moreover, 3D structures of the biological molecules can be analyzed through this application. When the organization and physical structure of entire cells is understood and represented in computational models that provide insight into how thousands of structures within a cell work together to create the functions associated with life. In any case, in the field of biology, structural bioinformatics is very important because structure alignments provide us with more information that is not available from current sequence alignment methods. Structural bioinformatics is now in a renaissance with the success of the genome sequencing projects, the emergence of high throughput methods for expression analysis, and identification of compounds via mass spectrometry. There are now organized efforts in structural genomics to collect and analyze macromolecular structures in a high throughput manner. These efforts include challenges in the selection of molecules to study, the robotic preparation and manipulation of samples to find crystallization conditions, the analysis of X-ray diffraction data and the annotation of these structures as they are stored in databases. In addition, there have been advancements in the capabilities of NMR structure determination, which previously could only study proteins in a limited range of sizes. The solution of the malate synthase G complex from E. coli with 731 residues has pushed the frontier for NMR spectroscopy and suggests that NMR is having its own renaissance. Finally, the emergence of this structural information when linked to the increasing amount of genomic information and expression data provides opportunities for linking structural information to other data sources to understand how cellular pathways and processes work at a molecular level. Protein structure prediction, protein-protein interaction prediction, molecular interaction and protein-protein docking are some of the applications of structural bioinformatics. All these can lead us to have better understanding in molecular biology. Two commonly used databases for the structural data are PDB and EMDB. PDB or Protein Data Bank. The worldwide website of the Protein Data Bank at the Research Collaboratory for Structural Bioinformatics (RCSB) offers a number of services for submitting and retrieving three-dimensional structure data. The home page of the RCSB site provides links to services for depositing three-dimensional structures, information on how to obtain the status of structures undergoing processing for submission, ways to download the PDB database and links to other relevant sites and software. EMDB or the Electron Microscopy Data Bank. In 2002, the EMDB was founded at the European Bioinformatics Institute specifically to archive the non-atomistic structures determined by different methods like single particle methodology, electron tomography and electron crystallography. Today, the EMDB contains over 1,300 released entries and is expected to grow 5 to 10-fold by 2020. Since 2007, the EMDB has been managed jointly by three partners: Protein Data Bank in Europe, RCSB PDB and National Center for Macromolecular Imaging or NCMI at Baylor College of Medicine. Functional Bioinformatics. Functional bioinformatics is nothing but the development and implementation of algorithms that facilitate the understanding of biological processes through the application of statistical and data mining techniques. Life sciences in general are generating huge amount of data with the accelerated development of high throughput technologies. Therefore, in parallel to the accumulation of the experimental information, there is the obvious need of analyzing all this huge amount of data. Data mining, text mining, gene expression and algorithm tuning are some of the main area under functional bioinformatics. Functional bioinformatics aims to define gene function by making use of the vast amount of information now available through high throughput experimental methods for mapping and sequencing genomes and approaches for characterizing genes function, the organization and expression under different conditions. Together, these functional genomic techniques contribute to the growing field of systems biology. These techniques are also being increasingly applied to understanding disease in human populations as part of genetic medicine. A particular disease characteristics and biomarkers for that disease can be explored through a gene expression analysis. It is a wide area in bioinformatics, and some of the main techniques used in this field are supervised and unsupervised methods. Unsupervised learners are not provided with classifications. In fact, the basic task of unsupervised learning is to develop classification labels automatically. Unsupervised algorithms seek out similarity between pieces of data in order to determine whether they can be characterized as forming a group. These groups are termed clusters, and there are a whole family of clustering machine learning techniques. But in supervised algorithms, the classes are predetermined. These classes can be conceived of as a finite set previously arrived at by a human. In practice, a certain segment of data will be labeled with these classifications. The machine learner's task is to search for patterns and construct mathematical models. These models then are evaluated on the basis of their predictive capacity in relation to the measures of variance in the data itself. Many of the methods referenced in the documentation are examples of supervised learning techniques. GEO and KEGG are the commonly used databases for functional data, even though there are plenty of other databases available. Gene Expression Omnibus or GEO. Gene Expression Omnibus is an international public repository that archives and freely distributes microarray, next generation sequencing and other forms of high throughput functional genomics data submitted by the research community. Kyoto Encyclopedia of Genes and Genomes or KEGG. Kyoto Encyclopedia of Genes and Genomes is a database resource for understanding high-level functions and utilities of the biological system such as the cell, the organism and the ecosystem from molecular level information, especially large-scale molecular datasets generated by genome sequencing and other high-throughput experimental technologies. Sequence bioinformatics. Genetic data represent a treasure trove for researchers and companies interested in how genes contribute to our health and well-being. Almost half of the genes identified by the Human Genome Project have known function. Researchers are using bioinformatics to identify genes, establish their functions and develop gene-based strategies for preventing, diagnosing and treating disease. A DNA sequencing reaction produces a sequence that is several hundred bases long. Gene sequences typically run for thousands of bases. The largest known gene is that associated with Duchenne muscular dystrophy. It is approximately 2.4 million bases in length. In order to study genes, scientists first assemble long DNA sequences from series of shorter overlapping sequences. Scientists enter their assembled sequences into genetic databases so that other scientists may use the data. Since the sequences of the two DNA strands are complementary, it is only necessary to enter the sequence of one DNA strand into a database. By selecting an appropriate computer program, scientists can use sequence data to look for genes, get clues to gene functions, examine genetic variation and explore evolutionary relationships. Bioinformatics is a young and dynamic science. New bioinformatics software is being developed while existing software is continually updated. The most important thing in sequencing is the identification of the gene within the given DNA sequence. Once the sequence has been assembled, bioinformatics analysis can be used to determine if the sequence is similar to that of a known gene. This is where sequences from model organisms are helpful. For example, let's say we have an unknown human DNA sequence that is associated with the disease prostate cancer. A bioinformatics analysis finds a similar sequence from mouse that is associated with a gene that codes for a membrane protein. It is a good bet that the human sequence also is part of a gene that codes for the same membrane protein. In such a way, the bioinformatics sequencing can be useful to find an unknown gene or protein of interest. There are bioinformatics programs like ClustalW, BLAST, etc. are available for sequencing. One of the widely used databases for sequence data is UniProt. The Universal Protein Resource, UniProt. The Universal Protein Resource is a comprehensive resource for protein sequence and annotation data. UniProt is a collaboration between the European Bioinformatics Institute or EMBL-EBI, the SIB Swiss Institute of Bioinformatics and the Protein Information Resource. Across the three institutes, more than 100 people are involved through different tasks such as database curation, software development and support. Let us now summarize what we have learnt in this session. If the genetic mechanism of a disease is known, then it might be easy for the doctors to take diagnostics tests more accurately. There are chances that in the near future, it will be possible to use genes instead of medicines to cure particular disease. In the future, it will be possible to cure or treat the disease by changing the expression of diseased protein with the help of bioinformatics. The identification of a gene expression level related with a particular disease is only possible with the help of bioinformatics. The integration of all the uses explained above can provide a clear picture about the gene. The structure, function and sequence are the important details about a gene, and all this information can be collected through a bioinformatics analysis. Without bioinformatics, new research in most fields of medicine and biology would come to a standstill. Here are a few questions for you to work out. 1. Describe the importance of bioinformatics. 2. What are the differences between functional and structural bioinformatics? 3. Write down the use of bioinformatics in sequencing. 4. Explain about different databases that use for functional data. 5. Try to figure out the common characteristics of functional and structural data. Here are a few books for your reference. Bioinformatics, a biologist's guide to biocomputing and the Internet by Brown SM 2000, Eaton. Bioinformatics, Tools and Applications by Edwards D. et al. 2009, Springer. Bioinformatics and Functional Genomics by Pevsner J. 2013, Wiley. Bioinformatics, A Practical Approach by ESQ 2007, Taylor and Francis. Bioinformatics, A Practical Guide to the Analysis of Genes and Proteins by Baxivanis AD and BFF Oluet 2004, Wiley. Here are a few websites for your reference: www.rcsb.org, www.ebi.ac.uk/pdbe/emdb, www.ncbi.nlm.nih.gov/geo, www.genome.jp/kegg, www.uniprot.org. Thank you for watching this program. Let's meet in the next session with a new topic. Till then, bye.